Hello everyone, today we will be talking about the biomolecules. Bio means life and molecule which form life are biomolecules. What are these biomolecules? These are building blocks, or we can say they are ingredients which make every living things. And these biomolecules are made up of non-living atoms like carbon, hydrogen, nitrogen, oxygen, sulfur and phosphorus. Together these non-living atoms combine in different proportion to form four major biomolecules, and these are carbohydrates exist both as monomer and as polymer, and they are energy source like glucose, and they also serve as a structural component like cellulose is present in plant cell wall and chitin which is present in fungal cell wall. Second one are lipids. Lipids are soluble in organic solvents like chloroform and methanol and largely insoluble in water, and they are classified as simple, compound and derived lipids on the basis of their chemical groups. So this is the example of simple lipids like triglycerides. So when you hydrolyze the simple lipids, you will get fatty acid chain. This is made up of hydrocarbon chain, hydrogen and carbon. Because of that, it is insoluble in water. It's also contain glycerol. So when you hydrolyze simple lipid, you get only two things, fatty acid chain and glycerol. So if you hydrolyze triglycerides, you will get three fatty acid chain and a glycerol. Compound lipids, if you hydrolyze the compound lipids, you will get fatty acid chain, glycerol plus a additional group like phosphate. In case of phospholipid, you will get carbohydrate as additional group in case of glycolipid, and you will get protein as additional group in case of lipoproteins. And derived lipids are obtained after hydrolysis of simple and compound lipids. That's why they are called as derived lipids. They perform wide range of function like they are a stored energy. Example triglycerides, they form membranes like phospholipids, and they are hormones like estrogen and testosterone. So these hormones are example of derived lipids. Phospholipid is example of compound lipid, and triglycerides are example of simple lipids. Next one are proteins. Proteins are polymers of amino acids. So when many amino acids combine together, they form proteins, and they play a structural role like keratin. It is present in our nails, hair and skin. They also play a functional role like hemoglobin. It carry the oxygen, and also play regulatory roles like insulin, which regulate the blood glucose level. Next one are nucleic acid. Nucleic acid are made up of polynucleotide chain. What is polynucleotides? When many nucleotides join together, they form polynucleotides, and these nucleotides are made up of phosphate, sugar and nitrogenous base. So there are two nucleic acids, DNA, deoxyribose nucleic acid, which store the genetic information, and RNA, ribose nucleic acid, which is involved in protein synthesis and gene regulation. In this lecture, we will discuss about carbohydrates. So why carbohydrates are important? Because they are most abundant biomolecules on the earth. And most of these carbohydrates are synthesized through the process of photosynthesis by plants. So when carbon dioxide and water react together in presence of sunlight, they produce glucose and releases oxygen. So glucose is the energy source, and it's used by many organisms like bacteria, fungi, protists, plants and animals. So where do we get it from? We get it from fruits, bread, rice, sugar and many other sources. They also serve as stored food. Like whatever extra glucose which is not immediately used for energy production, they are converted to their storage form like starch in plant and glycogen in animal. So these are actually extra glucose which are not used up immediately. So plant store this starch as tuber like potato, and as seed like corn, wheat and rice, and in a stem like sugarcane. And glycogen is stored in animal liver, like this is the example of human liver, and this cell is a hepatocyte cell. And if you look here, this cytosol contain this small purple granules, and these granules are glycogen granules. So whenever we require energy, this glycogen granule converted to glucose, and cells use that glucose for energy production. They also serve as structural components like cellulose, which is present in plant cell wall, chitin, which is present in fungus cell wall. Like this mushroom example of a fungi, and its cell wall is made up of chitin, which provide rigidity and a strength to the cell. And they are also, chitin is also present in exoskeleton of arthropods. For example, this grasshopper, this tiny exoskeleton of grasshopper is made up of chitin, which provide protection. And you can see many insects surrounding you, and its hard exoskeleton cover is made up of chitin. And last one are deoxyribose. So this is deoxyribose sugar, which is part of our DNA. It is the structural sugar present in our DNA. What are carbohydrates? They are hydrates of carbon. This is made up of two words. Carbo means carbon and hydros means water. Water is made up of hydrogen and oxygen. So basically carbohydrates are made up of three major atoms like carbon, hydrogen and oxygen. They are also named as saccharide in Greek, which means sugar. This name was assigned to carbohydrate because of their sweet taste. But all carbohydrates does not taste sweet, and they are represented with the empirical formula of C dot H2O whole N, or it can be written as CnH2O whole N. Example is glucose. Glucose has the molecular formula of C6H12O6, or it can be written as C dot H2O whole 6. But all carbohydrate does not fit in this formula. For example, deoxyribose, which is present in our DNA, it has the molecular formula of C5H10O4, and it lacks one oxygen to fit in this formula. Similarly, all molecules which follow this formula are not carbohydrate. Example is acetic acid. It has the molecular formula of CH3COOH. It can also be written as C dot H2O whole 2. But it is not a carbohydrate. Some carbohydrate apart from these atoms like carbon, hydrogen and oxygen also contain nitrogen, phosphorus and sulfur. So accurate definition for the carbohydrate is they are either polyhydroxyaldehyde or polyhydroxyketones. Let's understand this with these two sugar molecule. So if you look at this sugar molecule, this is 6 carbon sugar. It has aldehyde group as its carbonyl carbon. What does carbonyl mean? When carbon is double bonded to oxygen, it is known as carbonyl. So it has this aldehyde group and many OH group, like here 1, 2, 3, 4 and 5. So they are polyhydroxyaldehyde. If you look at this sugar molecule, it has carbonyl carbon as ketone, and all these carbon are attached to 1, 2, 3, 4, 5 hydroxyl group. So this is polyhydroxyaldehyde and this is polyhydroxyketone. So what are carbohydrates? Carbohydrates are either polyhydroxyaldehyde or polyhydroxyketones. Now let's see the classification of carbohydrates based on the number of sugar units. So first one are monosaccharides. Mono means single and saccharide means sugar with single sugar unit, and they are the simplest carbohydrate because they cannot be further hydrolyzed to produce another carbohydrate. Examples are glucose, fructose and galactose. Second one are disaccharide. Di means 2 and saccharide means sugar. So when two monosaccharide combine together, with glycosidic bond, they form disaccharide. Examples are maltose, sucrose and lactose. Third one are oligosaccharide. Oligo means few. So when 3 to 10 sugar units combine together, they form oligosaccharide. Example are maltodextrose, it's made up of 3 glucose unit, and raffinose, it is made up of 3 sugar units, and these are different sugar, one of galactose, one is glucose and one is fructose. These 3 together combine to form raffinose. So maltodextrose and raffinose are example of trisaccharide. So now the last one are polysaccharide. Poly means many and saccharide means sugar. So when more than 10 sugar units combine together, they form polysaccharides, and example are starch, glycogen and cellulose. Now we'll see the classification of monosaccharide. Let's first see what are these monosaccharides. Monosaccharides are colorless crystalline solids and they are soluble in water. Most of them have sweet taste. Not all of them, but most of them have sweet taste. So they are classified on the basis of number of carbon atoms present in their backbone. So these are between 3 to 7. So if they have 3 carbon atoms, they are known as triose and they are represented with this suffix OSE, O-S-E. So if they have 4 carbon atom in their backbone, they are tetrose. With 5 carbon atom, they are pentose. With 6 carbon atom, they are hexose. And with 7 carbon atoms, they are heptose. They are also classified on the basis of functional group they have. So if they have functional group aldehyde, means carbonyl carbon. If it is aldehyde, they are known as aldose. If it is ketone, they are known as ketose. So we can combine both of these convention to name them. So like this sugar is a 3 carbon sugar with aldehyde group, it is called as aldotriose. Similarly, this 3 carbon sugar is with ketone group, it is known as ketotriose. So this is the structure of glyceraldehyde, this is the structure of dihydroxyacetone. So glyceraldehyde is a aldotriose and dihydroxyacetone is a ketotriose. Next we will see another example. This is 4 carbon sugar with aldehyde group. It's a aldotetrose, and this is 4 carbon sugar molecule with this ketone group is ketotetrose. So this is the structure of erythrose and this is the structure of erythrulose. Similarly, with 5 carbon atom with aldehyde group, this is aldopentose and this is the structure of ribose. This is with ketone group, this will be ketopentose and example is ribulose. And 6 carbon sugar with aldehyde group is aldohexose and this is the example of glucose. And with 6 carbon sugar with ketone group will be ketohexose and this is the structure of fructose. So glucose is a aldohexose and fructose is a ketohexose. All these were the examples. Likewise, carbohydrates are named. Monosaccharides are named. Now we'll see the structure of monosaccharide. So this is the acyclic or open chain structure of monosaccharide, and we draw this by using Fischer projection. So this structure is called as Fischer projection. This is the structure of glucose. So we first place this carbonyl atom, that is aldehyde group, then you move downward and place the other carbon atom. All these vertex represent a carbon atom, and this all carbon atom are attached to either hydrogen or hydroxyl group, both the sides. Now in aqueous solution, sugars prefer to be in their 3D chair conformation. So if you look, this is the cyclic structure of glucose. So when you draw this vertical axis here, you will see these all groups, this H, OH, H, H and this H, all are in perpendicular to this axis. So these groups are called they are at axial position, and whatever groups, these are slightly perpendicular to this axis, like this OH, OH, this OH, this OH and this CH2OH, they are known as, they are placed at equatorial position. So at equatorial position, steric hindrance between the groups are lesser. So equatorial position of bulky groups are more favorable. So when you look at this structure, these all heavy groups like OH, 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 OH and CH2OH are in their equatorial position. So this is the most stable structure of glucose because these groups have less steric hindrance to each other. This is the Haworth projection. Here we use the Haworth projection to draw the cyclic structure of the sugar. This is but in real scenario, Haworth projection is not possible because all these groups are represented in same plane, and it's not possible because of the steric hindrance between the groups. So we use Haworth projection just for because it's easy to understand and draw. So whenever you see the cyclic structure of glucose, you mainly see this structure, but in aqueous solution this is the most stable structure of the sugar. Now we will see the cyclization of sugar. So this is the open structures, then how cyclic structure is actually formed. So this is the basic chemical reaction between aldehyde and alcohol which form a hemiacetal group. So actually this is the hemiacetal formation. So if you look at this fifth number of this OH group, this has a lone pair and this attacks this aldehyde carbon, and when this attack this aldehyde carbon, this oxygen form the bond with this first carbon and this O is converted to OH. Here similar like this carbon has initially had this R group, this H group, and this O is converted to OH and this group another OR. Now let's see this. So I have represented this in two different form. You can draw this way or you can draw this way. So let's understand this. Since this O attacked this lone pair attacked this carbon, this O will be formed between this fifth and first carbon atom. This cyclic O is formed. And final structure, and here this OS is formed, and final structure will look like this. You can draw this way as well. You first place this first carbon, carbonyl carbon, and then move to clockwise direction. Place the second carbon, third, fourth, fifth, and the sixth will be here. Why 6 will be here? If you drawing the D glucose, this 6 will be CH2OH will be upside. If you are drawing the L glucose or L sugar, this will be downward. And now it's very easy to draw. Whatever on the right side of this structure, you put them downward, like OH, OH, H, OH, you place here OH, OH, H, OH, and whatever on the left side, you put them upward, like H, H, OH, H, H. So H, H, OH and H. This way you can simply make the cyclic structure from the linear structure. So if you remember the linear structure, it's very easy to draw the cyclic structure. This is the another type of representation. So you can make first this structure, place this lone pair here, and this will attack here and make bond between them. This O is now become the cyclic ring O, and then this O is converted to OH. So you must have heard the term of alpha glucose and beta glucose, and that are because of this position of this OH group, newly formed this OH group. If it is in downward direction, it will be alpha sugar. If it is in upward, here upward position, it will be beta form. I have not shown here because alpha and beta form are isomers, so I will talk about them when we will study the isomerism in carbohydrates. Now look at this structure. This is a 6-member ring chemical compound, and it is known as pyran. And if you look here, this sugar molecule look exactly similar to this molecule. It has 6 ring with ring oxygen. Similarly, it has 6-member ring with ring oxygen present here. So we can name them sugar, name the sugar like this. Since this is a structure of glucose, we can name it D-glucopyranose. Similarly, this is the structure of fructose, cyclic structure of fructose. So this fructose has since ketone group here, it is present at second position. This lone pair of oxygen attack this carbon and form the bond between them. This oxygen will become the ring oxygen. So here is the structure of this fructose. So why how this happen? When ketone and alcohol reacts, they form hemiketal. In case of glucose, it was hemiacetal because alcohol was reacting with aldehyde. Here alcohol is reacting with ketone, so it will form hemiketal group, and finally fructose will cyclize. Now why ketone is at second position, not at first position? So it's simple chemistry. So if you look at here, if you place to be at first position, here must be a hydrogen. If it is a hydrogen, it will become aldehyde. If you place you will replace both the R with hydrogen, it will become formaldehyde. So then you need to keep the ketone always at second position because it attached with two different R group, and this R cannot be a hydrogen atom. So now if you see this structure, this look very similar to this five member ring cyclic compound, and this is known as furan. So fructose can also be named as fructofuranose. This way alpha and beta fructose are also formed because of position of this OH group. If this OH is on downward side, it will be alpha. If it is on upward side, it will be beta fructose. These are the few common monosaccharides. These are glucose, fructose and galactose. And glucose is present in rice, bread, fruits and honey. Fructose is present in fruits, honey and table sugar. Galactose is present in milk and milk products. If you can see, these all three have the same molecular formula of C6H12O6, but they are different. They have different physical and chemical properties. Why this is happening? Because of isomerization. So in next lecture, we will discuss about the isomerization in carbohydrates. Other very important monosaccharides are ribose sugar, which is present in RNA, and deoxyribose sugar, which is present in our DNA. So if you look at the structure of this ribose sugar and this deoxyribose sugar, they have only one difference at this second number carbon. In case of ribose sugar, this second number carbon has a alcohol group, OH group. But here this is not OH, it's H only. So since oxygen is lacking at second number position, this is called 2-deoxyribose. So this was all about the monosaccharide. Thanks for watching. See you in the next video. In next video we will discuss about the isomerism in carbohydrates.